Welcome to the Broad Stem Cell Research Center at UCLA. We will now walk you through the key findings of our recent study published in Developmental Cell, which describes how the trophoblasts regulate the placental hematopoietic niche through PDGFB signaling. Understanding how the microenvironment or the niche directs the fate of hematopoietic stem and progenitor cells is a major goal in our field. During embryonic development, hematopoiesis is segregated into multiple anatomical niches that either dictate the generation of differentiated blood cells for the embryo or promote the establishment of a pool of undifferentiated hematopoietic stem cells for postnatal life. However, the cellular and molecular components of these different niches remain completely undefined. In our previous work, we discovered that the placenta is a unique hematopoietic site that harbors a large pool of hematopoietic stem cells that are protected from differentiation. We then identified two distinct vascular niches in the placenta: the large blood vessels in the chorionic plate that generated multipotent hematopoietic stem and progenitor cells, and the placental vascular labyrinth that promotes HSPC expansion.
In order to understand how the hematopoietic niche in the placental labyrinth is constructed, we studied mouse embryos deficient for PDGFB signaling. We chose to study this pathway as loss of PDGFB or its receptor tyrosine kinase PDGF receptor beta results in a compromised placental labyrinth structure. This includes dilation of the fetal vasculature and reduction in trophoblasts that line the maternal blood spaces. We discovered that in the absence of PDGFB signaling, the placenta starts to support definitive erythropoiesis, which should only occur in the fetal liver at this stage. The mutant placentas harbored large clusters of highly proliferative erythroblasts in the placental vascular labyrinth. The erythroblasts in the mutant placentas were clustered around central macrophages, forming erythroblast macrophage islands, highly reminiscent to those found in the fetal liver. Our study revealed that PDGFB ligand secreted from the endothelium activates PDGF receptor beta on trophoblasts to suppress secretion of erythropoietin, the main cytokine promoting erythropoiesis. We showed that the ectopic erythropoiesis can be induced in the placenta upon loss of the ligand, loss of the receptor, or overexpression of EPO, specifically in placental trophoblasts.
This study is the first to identify placental trophoblasts as powerful signaling centers in the placental hematopoietic microenvironment. Our work also reveals a completely new role for a widely studied signaling pathway, PDGFB signaling, as a hematopoietic niche factor that regulates EPO levels and thereby restricts the differentiation of fetal hematopoietic stem and progenitor cells to appropriate niches. We believe that identification of the niche cells and signals that dictate the balance between stemness and differentiation will ultimately enable us to develop culture protocols to exploit the full potential of stem cells in regenerative medicine.